Our primary goal during this symposium is going to focus, of course, on the domestic cat, and nearly all of tomorrow will be focused on that. But domestic cat, of course, among other things, is in the context of being a cat. And cats are more than just domestic cats, and so most of this evening is going to be focused on the non-domestic cats, the larger and wild cats. And it's a great pleasure to now begin with our first speaker. And Warren Johnson comes to us from the Smithsonian Conservation Biology Institute as a research scientist. He received his PhD in ecology from Iowa State, and he joined the Genomics Diversity Lab at the National Cancer Institute in 2000. His research has spanned a broad spectrum of disciplines and comes to us specifically as the expert on cat taxonomy. In fact, as you know, taxonomic relationships are going through revolutionary changes right now because of the ability to use genetic information to supplement anatomical and fossil information. And all of us want to know what our cat cousins are and how many genera there are and so on. Dr. Johnson.
First off, it's a pleasure to be here tonight and to see such a full room. I thank Al and Nora Gress, as well as all of the other sponsors. This promises to be a very exciting and interesting conversation, or dialogue, as it was proposed to us. And I'm going to attempt to start that dialogue. And that, although the title has the word taxonomy in it, I think taxonomy is, as you will see, really just the smallest tip of the iceberg. And what I hope to really do is put this whole thing of evolution in context, not just cat context, but mammalian context, so to speak. So, but just a brief introduction of who I am and where I came from. Actually, animal behavior was what brought me into this realm. Although after spending several years studying coyote behavior and social behavior in small pups at Utah State University, and then going to the Patagonia of southern Chile to study puma and mountain lion behavior, I began to realize there's only so much one can do with a pair of binoculars. Even the best attempts to replicate something in animal behavior is almost a non-starter, and stumbled upon really genetics and its role in all of these things. And I think it's going to be perhaps the one thread that's going to go through a lot of what we're talking about here. From there, I really learned my genetics, not doing the field work. That was basically a field ecology study, but at the National Cancer Institute and the Laboratory of Genomic Diversity under Dr. Steve O'Brien, who many of you may well know, and spent a lot of time collecting the samples and the other material that one needs to be able to do these sorts of studies. So today I'm going to talk quite a bit about felid evolutionary history, and then the process of domestication that brings us to what we're here for, the domestic cat in many ways. But then I'm going to broaden it right back out and touch on other things that the genetics of domestic cats can enrich us with, health and behavior, but then really bring it into a further context of comparative genomics and conservation and many of the other things that we will also be talking about over the next day and a half. A lot of what I will be presenting is summarized in this paper, State of Cat Genomics, and several of those co-authors are intimately associated with much of what I will present as well. So if there's one thing you should look for afterwards, if you want to find another place, it's here. The cat evolution story is also been described in a National Geographic study as well. In context, a lot of mammals, over 5,000 species. Mammals taxonomy, so to speak, or evolution, has really undergone a sea change over the last, say, 5 to 10 years in terms of how we organize our thoughts about mammals. But there really are essentially 4 main groups of mammals, which are outlined here. And The ones that we're interested in today, these carnivores, share a group with other species, the ungulates, the bats, cetaceans, and hedgehogs and moles. And you'll see the implications of this in a minute. I'm going to show quite a few, or depict evolution in these phylogenetic trees very often. And in this case, this phylogenetic tree has contemporary tips here in an attempt to actually look at the depth of evolutionary time, so to speak. And what you will appreciate from here, the KT boundary, if you remember, this was a period of time when there was a lot of change, and the main portion of that change were the dinosaurs were being replaced by many other species. But the important part of this story here is that the precursors of most of the mammals that we see today already were running around, so to speak, within the same habitat and sharing the same places as most of the dinosaurs. But what you don't appreciate from this depiction that it wasn't until the KT boundary when many of the species and the lineages that we see today actually radiated and diversified, so to speak. So where do the cats come from? Well, I had to put in my own dog and my own cat here. For those connoisseurs, there's a Labradoodle, a pound cat, very pretty, nice one. But just so we start thinking about evolution and genetics, who would have guessed that the closest relative to all carnivores is something called a pangolin? Many of you probably haven't even heard the word pangolin. It happens to be the species that is most traded in the world for both meat and for medicinal properties, both in terms of tons, actual physical weight, and individuals. Old taxonomists used to group these with armadillos, simply because they have scales, they don't have teeth. So here we go, the immediate ancestor to all carnivores. The hair has turned into scales, the teeth have disappeared. So if you look farther out in evolution, all mammals tend to have teeth, right, for the most part. So in these species, these genes, this genetic, it's not like they've lost those genes in many cases, they've just lost that function. In the meantime, as we approach this lineage of carnivores, they're retaining features, of course, that they share, but then as they diversify, they're getting features which make them unique, and which we're going to spend a lot of time talking about. So the question posed, where did the domestic cat come from? Well, there are today anywhere, depending on how you might count them, 37, 38 species of living cats, roughly distributed in different ways around the world, and we'll visit that a little bit. Most of this came out in a science paper a few years ago that looked at this using the molecular markers of that time. So it's not that this question hasn't been addressed before. In fact, it's been of real great interest to taxonomists really since Linnaeus and others started trying to group animals together. But if you think about, if anyone's looked at a cat's skull, for instance, from one species, from a lion, say from a domestic cat or a lynx, there's very little to distinguish it except size. And it turns out that's the case for almost all measurable features, just looking at skeletons. Well, it turned out to be the case for other things, such as chromosomes, for some of the old genetic methods that were used, albumin immunological distances, protein electrophoresis, mitochondrial DNA. And it wasn't until recently that we actually had the tools and the samples from all of the species to actually begin to address this. So the strategy here was to sample all cats, outgroups. The reason it's important to have the outgroup, that allows you to help establish which are the older lineages relative to the other ones, and then several nuclear genes. And this is a complex phylogenetic tree, so to speak, but it's simply here to depict a little bit the pattern but mostly that to show that there are 8 evolutionary lineages of cat species. Many of these were quite a surprise. Many taxonomists had actually placed the cheetah, has some very unique features, right, that doesn't share with any other cats, as the most ancestral, the oldest cat lineage. Turns out the cheetah is actually right there in the midst of a group with the puma and the jaguarundi. Surprise. We can now explain probably why that happened. So I'm going to go through some of these details here. But you can, as you look at this, though, you can see that, remember, branch lengths tend to represent the amount of time. So immediately, we're seeing that these branch lengths among these groups are all very, very short, trademark of a very rapid and quick radiation. We can put dates on these things. So it turns out that although these species can be hard to distinguish and group, there are lots of fossil records that we can use to start trying to figure out what are the actual dates that these events occurred. And when you do that, it tells us that all modern cats essentially share a common ancestor roughly 10 million years ago, 10 to 11 million years ago. After that, really in an evolutionary sense, during a very short time period, they radiated into each one of these groups. So can we explain what processes went into that? Is there a simple story? Well, it turns out there is. This is a recreation of what that ancestral cat might have looked like. And what we're seeing is that the radiation of modern females probably started in 10 to 11 million years, Asian. And the first groups to diverge were Neophelis, that's the clouded leopard. All of the Panthera, those are the big cats, those charismatic ones that we're so interested in, and this Pardofelis, followed what really is a relatively simple biogeographical story. And this story is related to different levels, sea levels. And you can imagine why. Speciation in general often happens when populations become separated from one another and then are allowed to change, to adapt to different time, to different different environments and other kinds of selective forces. So a lot of these lineages are associated with times when sea levels were high. High sea levels tended to separate continents, right? Whereas a lot of the tips of these are occurring during these rapid periods of sea level change. Again, isolating some of these populations. And we'll go through a few of those examples in a minute. So during the Miocene, during those early time years, remember the precursors are here in Asia. So the first set of migrations occurred 8 to 9 million years ago into Africa. And the precursor of the caracal lineage. So this is a group of all African species, 3 African species. The second set of migrations would have occurred when the Bering Strait was connected. So that was the first time that North America actually experienced felids. And these are the precursors of the puma, the lynx, and the ocelot lineages. And when North and South America connected, roughly 2.5 million years ago, this is when the ocelot lineage came into South America. And this is the only species which is unique to South America. All members of this group entered, the precursor entered, and they diversified into this group. Happens to be the only group that also has a different chromosomal conformation from all of the rest of the cats. Happened during that time period. More recently, the cheetah, which was a North American species, and there are North American fossils, went back into Europe. A precursor to the lynx went back into Eurasia, I should more correctly say. The cheetah then made its way into Africa and subsequently, won't be part of tonight's talk much, but went extinct and is actually found still in parts of Eurasia. It's now essentially an African species. Black-footed cat, it's one of the the domestic cat lineage species, which we'll talk about a bit later. The Panthera genus, lions, went into Africa. Lions also went into North America. In fact, there are lion fossils in South America. And then, of course, then they were extirpated, just as cheetahs were. Pumas and jaguars, so remember jaguars are pantheras. They're big cats. They came down into South America and are essentially found only in South America relatively recently. And we'll come back to it in a bit, but there were even examples then of more recently animals coming back out of South America and vice versa. That's all of which we won't have lots of time to talk about today, but which become very important when we start considering modern distributions and when we start trying to link what we see today, especially in terms of conservation, with the evolutionary history. So I still haven't actually gotten to what we're here to talk about, the domestic cats. And this is where I introduce, this is now Dr. Carlos Driscoll. At the time, he was doing his PhD work on this. And as with all genetic studies, he spent almost a good proportion of his first couple years of his PhD work collecting the right samples. If you don't have the samples from the distributions of the cats, and if you don't have the samples of the non-domestic cats, then your chances of solving the puzzle, no matter how hard you try, are going to be in vain. So there were several candidate species here. We had lots of good guesses. And essentially, it won't be of any surprise to you, Felis sylvestris, the wild cat, and generally recognized as 4 subspecies of these: the Asiatic, the European I should say, the Asiatic, the Southern South Africa one, and a Near Eastern one, Felis sylvestris libica. And they all look a lot like domestic cats, don't they? In fact, if you go into the areas where these wild cats are found and you see a small cat, usually you will misidentify it. It's not a trivial process. People have been thinking about domestication for a long time, so it wasn't as if there were no theories out there. So the best guesses really were that domestication was a Middle Eastern, Mesopotamian event, roughly 4,000 to 8,000 years ago, based on records and paleontological evidence, so to speak. It was presumably a single event, so it happened once in one area, and that was a hypothesis that could easily be addressed using the genetics. The curious mechanism, we'll come back to that in a minute, but unlike other species, it was pretty clear initially that the domestication perhaps wasn't human initiated as, at least in its early stages. Certainly, there was irrefutable evidence, and both from records and others, of when domestic cats were found and started to be kept. And that's some of that evidence is here, and others will go through that evidence later. So really, the key to solving the problem really is to get the right samples. So Carlos went out, and lots of colleagues and collaborators looked for the putative wildcats. And of course, I'm saying putative because we don't really know what the wildcats are. We don't know if they're pure. If morphologically, if you look at them, they look a lot like our domestic cats. Feral cats, it's hard to tell sometimes whether they're wild or domesticated, as you know. And using at the time the best markers available, 36 microsatellite loci, they're mapped so we knew where they were in the genome, and several mitochondrial genes. Mitochondrial genes trace the maternal lineages, so that's tracing female to female to female. That'll be important as we go through some of the results. This is microsatellite data. It's nuclear data. Microsatellites are neutrally evolved, so they're not necessarily selected for, but they have this nice trait that they evolve quickly. So therefore, they allow us to look at events that have happened very recently. And that's important, of course, because domestication occurred recently. This is another way of looking at phylogenetic tree evidence. Essentially, tips that are joined together are more closely related to each other, right? And distance has meaning here, both in the internodes, but in the tips. And in this depiction, color also has a meaning. So all of these lines here represent an individual cat. And all of these lines in this color represent the individuals that were found in the Near East, or were initially identified as a domestic cat. So I'm giving you the punchline in some way, to a large extent. These are the Middle Eastern cats and domestic cats from throughout the world. These are European wildcats. These are the South African wildcats, Central Asian, a Chinese desert cat, which is essentially wild but falls so close in within these domestic cats that many of us actually believe it is another type of a wild cat, so to speak, here. I think I misspoke. It's nested within the wild cats. And then the natural outgroups of these wildcats are firmly recognized as other species. But you'll see here in tips, and I'll come to this in a minute, each line that has one of these circles actually has a mitochondrial marker that tells a different story, that actually looks like a domestic cat. And I'll show you that in a minute. So what does that imply? This implies that these individuals are hybrids, so to speak. I don't necessarily want to use that word, but there's introgression. Part of their genomic background is domestic, or from this group, and part of it is from one of these other groups. So it's a complex story. If we try to put it onto a geographic map, you'll see in green are the European wildcats, and addition, you can go across the geography. But what to note here is that the Near Eastern wildcats and the domestic cats are all falling within this microsatellite group. And so that's where the European wildcats are. So that puts the microsatellite data on there. And so superimposed on top of that is the mitochondrial data. And those are in the circles. So mitochondrial, you have haplotypes. You inherit the mitochondrial altogether, so you have what's called a haplotype. And so there are going to be very many fewer of these. So the take-home message here, to simplify it, is that each haplotype, in this case, has a number, and they've been grouped by where they were found in terms of the wildcats. So in this case here, all of these cats that were found there either had a haplotype that was only found in the southern part of Africa, or it's brownish haplotype that was found from here and happens to be found predominantly in domestic cats. And if you look around, you'll see that that brown haplotype's found all over. It's not found in as high frequencies elsewhere, but it's also the one that's predominantly in the domestic cat. So what's it telling us is that this is consistent with the fact that those mitochondrial lineages, and most of those mitochondrial lineages we see today in the domestic cats, are from this region. And that all of these haplotypes either extended in some way in the wild cats, or as probable, came from introgression from domestic cats breeding with the local wildcats. Okay, so another way of showing this, in this case, this is back to the microsatellite data, nuclear data. Each line, of which there's so many that sometimes you can't see the individuals, is representing an individual. Colors are representing essentially relatively recognizable and homogeneous groups. So if we look here, there's the European wildcat nuclear grouping, so to speak, South African, Asian, the Near East, and most of those domestic cats, again, Asiatic domestic cats, Mongolian domestic cats, European domestic cats. We can recognize this ancestry, but again, it's very consistent with a Near East source of domestication. So to sum all of that, as easily as we can, the process of domestication seems to have happened in a region of the Fertile Crescent around the time of agriculture. And I didn't show you how we dated this, but the evidence is that at least 5 mitochondrial lineages, much older, say about 100,000 years ago, are found in modern domestic cats. And so this suggests that the process of domestication, or that period of time when there was lots of gene flow between that domestic stock that was forming and the wild stock, is much older than what we would say from the archaeological record, say domestication occurred. But it does point that it was in this part of the world, the Fertile Crescent, probably in a relatively small area, and in the same area, parenthetically, where a lot of domestication occurred. And this is a list of some of the other plant and animal species that were domesticated in this region. It's a period of time when humans became less nomadic and started settling down. And with that process of starting agriculture, et cetera, started cohabiting with mice. And so the theory goes, the cats started coming towards this easy source of food. The humans, of course, didn't mind, didn't complain. And slowly, selection occurred. Started seeing this actually happened much later, but in a process which in some ways is different because perhaps cats domesticated us as much as we domesticated them in some ways during that time period. So it's a different story of domestication, perhaps, and I think it's reflected, and we'll hear in how we perceive and interact with our cats. But an important part of understanding cats as we see them today, and domestic cats and domestic cat breeds, is that unlike dogs and other breeds, the cats and cats breeds are relatively open in the way that they're managed, so to speak. And you're now experts, right, at looking at microsatellite data and how they get grouped. And this is a structure plot. And this captures that a little bit. If you were to look at a similar thing like this, these are different breeds here across this line. So these samples have been collected from the 38 main cat breeds. Each line represents a different individual. In some cases, we've redone the analysis with small groups here. But what this story really tells us in some ways is that these cat breeds have been interbred sufficiently and selected for certain traits sufficiently so that even with just a small number of microsatellites, we can recognize their heritage, their genetic heritage. But as I was beginning to say, if you were to do the same exercise with dog breeds, you would get patterns much more like this, where you would have very strongly defined and recognizable groups. Most dog breeds are closed breeding processes, and that happens with much less frequency among cats, as you know probably better than I do. And so that's reflected by the fact that you get lots of different colors. Each one of those colors, in essence, is representing that mixed heritage. So I'm now going to transition over. We can talk a lot about taxonomy. In fact, I didn't talk any about taxonomy. Taxonomy is a way that we help communicate with each other and provide names to it. I put the names up there, and we can certainly spend some time talking about it. The cat breeds, most of you may well be familiar with. But what I think is as interesting is to take that structure. I mean, taxonomy is a way of trying to put words on genetic structure, populations, or shared history, or whatever. And what can it teach us about what has happened, but as much as take advantage of it for the kinds of things that we would like to do? And that process has become much easier and much more accessible for everything, and I hope to give you that flavor. The cost of computing Moore's law, many of you have seen this, is dropping. If you look at the cost, though, of sequencing genetic data, it's broken all of those laws. It's now essentially for only a few $100s, we can, for each one of us in this room, learn what the important variations are between any one of us in the room. And it's already seeping into our lives in many, many ways. So this genomic era is really offering us the opportunity to use the relationships between genetic features and function to actually look at what's different among organisms and then compare those. Take all those evolutionary experiences and experiments, so to speak, and link them with genetic tools. And so Dr. Steve O'Brien in our lab really started pioneering some of these things, this comparative genomics projects where we took whole genome sequencing, put it into an evolutionary context, and then started looking at these natural experiments. So the first, the domestic cat, was actually one of the pioneers in getting the whole genome. And why is that important? I'll show you that in a minute, but here's the citation and the authors of that process. It was sequenced initially at a 2x coverage. That simply means today most things are sequenced at 8 to 10x coverage. It gives you a sense for the depth and breadth of the information. Cinnamon, and it's important to know the history of the cat that's being sequenced. We'll come back to that. The heterozygosity in this cat was actually relatively low, and they chose that at the time on purpose because it made it easy to put all the pieces together, because less variation. But it was also part of a population that was being line bred, so to speak, because Cinnamon is an affected cat from a retinal atrophy pedigree maintained by Christina Nordstrom. Cat's already been upgraded, and sometime in the next year you'll see a full 10x coverage of Cinnamon, and then in a lot more genomic data that I'm not going to go into today, but for those of you that are interested in getting early access to some of that, please talk to me, and I can put you along help you get access to that. But why are we interested in these things? So one thing you can do with all of this genomic data, and one thing we're interested in, is we all know the kinds of diseases, the kinds of phenotypes that are segregating in cats, and this list is much longer. It was put together by Urs, and that can be found at this site, of some of the phenotypes, some of the disease, genetic associated diseases that are found in cats. Increasingly, an increasing number of these, and this has not been updated over the last couple of years, have now even been characterized at the molecular level. So already, and just to give one example of that, of many, is the spinal cord disease here that has a similar disease or phenotype in humans. So the implication here is that this deletion in feline spinal muscular atrophy is not just allowing us and giving us tools to understand this disease, this cat evolution, but then tying it back to human stuff. And why is that important? Well, here's another list of examples. Colors. Colors have fascinated geneticists for many years. We now know the causative mutations for many of these phenotypes, including some of the wildcats. And not on here, for instance, if you're familiar with king cheetahs, the genetic mutation for the phenotype of king cheetahs was, just came out in press in the last month as well. But not just diseases. Many of the viruses have human homologs. FELV, FIV, and here's the human correlates, associate. Coronaviruses, oncogenes. Oncogenes were at one point thought to be highly associated with cancers. Flus, panleukopenia. I won't spend a lot of time talking about that, but if you're interested, I can certainly help point you in the direction. And parenthetically, it were these features that actually got Steve and NIH interested initially in the cat as a model. So why are we interested in genomes? The other way to think about genomes are genomic maps, they're sometimes referred to. There are ways to order in a genetic way all of this genetic information. Well, it's this framework, so to speak. Well, it's really important, it's helpful to both track the evolution of the chromosome itself, but it also gives us the ability to compare all of this genetic data. So if you have a phenotype in a cat that you also see in a dog, how do we go about studying that? Well, if you know the genetic landscape for your markers, that's often a very useful help. So I've been working a lot with the alpaca and developing a genomic map of the alpaca with others here. So if I find a gene of interest in a mutation, what I'd really like to do is say, well, what else is known about it? What is known about it in human, for instance? And so I could look at the sister groups of dog and cat. In this case, I've also put up the horse here. Those are the genes of interest. And so where are they found in the dog and cat? Oh, look at that. Dog, cat, and horse. Turns out that the synteny, the order of all of these genes, and this is, are similar in these species. And so what does that do? That tells us that a genetic marker or a gene in one species is going to be very similarly located in the others, and that will help facilitate you finding that correlation between the marker and the gene, and then to start looking at other features about those. So comparative genomics, the relationship of the genome, it can be across species, it can be across strains, it can be at multiple evolutionary levels. A lot of genomes now that have been done at high coverage, most of these, if you look at them, are lab models, but increasingly they are agricultural species of importance. But through organizations such as the Genome 10K Project and others, increasingly there are also species that have conservation interest or just evolutionary interest. The costs are coming down so fast. Well, if you have a lizard you want to know something about, now you have ways, if you do a genome, to learn about, to figure out, well, what genes are most likely to be interesting for a certain phenotype. So I keep using this word phenotype. It's important that we understand that because we're going to talk a lot about phenotypes in the next most of the day. We're going to, I think I'm the only genetics talk. Phenotypes cover a whole host of things. They can be morphological, all that is entailed, right down from coat color to whether you have three fingers or 4 fingers, or the way you actually might respond to certain things. Coat colors, behavior is a phenotype. Very often, the way you behave has a genetic component. Disease resistance, life history traits, whether you breed once a year, twice a year, what time of year, have known genetic correlates and differences. And these differences often are found within and among populations. So if anything that's heritable, anything that you can pass on, essentially, is likely to have a genetic correlate, and that we can start talking about what the genetic cause is going to be. I promised that there would be some conservation applications here. Certainly diversity, how much variation. Evolution can't operate if there's no variation, right? So cheetah's been the sort of the poster child of that. Very low genetic variation, but somehow survived all of that. But in order for the cheetah to evolve and change, it's going to have to generate more variation. So maintaining variation is important, and we'll discuss, I'm sure, why maintaining variation in breeds becomes important. Taxonomy and systematics, what do we preserve, what do we conserve? Hybridization, phylogeography, gene flow among these. You could substitute these same things among domestic animals, right? If you have closed breeds with no gene flow, lack of variation, going to be concerns. Behavioral ecology, how do we tell who's related to whom? We can date when these breeds happened, when these events happened. And then we can start looking at all of the biomedical implications. Breeds that have high incidence of certain good traits and bad traits become interesting. And we now have the tools to address that. And then gene mapping and forensics speaks for itself. We can look at that. Real quick, I'm going to give some examples of these. The Iberian lynx, who would have thought the world's most endangered cat is actually in Europe? Actually mostly in Spain. But already they are maintaining this species alive mostly in large extent based on technologies that were developed in humans and then translated to cats, which are now being translated to other species, and reintroducing them into the wild. This is a North American species, very common throughout, and we're just now beginning to understand what kind of substructure and what kind of adaptations there may be to one population or other. Put this also to remind us that domestic cats aren't so highly refined, so to speak, that they don't still interbreed with many of their older ancestors. And one of the questions we get all the time, over and over again, is, can you tell us how much of a bobcat is in my cat? Right? Is it 20%, 40%, 60%, or any? So now we have tools. We can start really doing a good job of answering those questions. The cheetah, the poster child, I put that up because it's a member of the puma group. The best example, really, of conservation genetics, if you're not familiar with it, is right here in Florida. And recently, a science paper came out that went through the last 20 to 30 years of that very large study. But essentially, it has all the elements of conservation genetics. Small populations, inbreeding, individuals that were beginning to not breed very well, disease transmission, hybridization, systematics, and then the decision to introduce new genetic material into that population in an effort to save it. Because it went for many generations in a very small population size, inbreeding, loss of genetic variation, but to the extent that everyone thought it was going to go extinct. Now, pumas weren't going to go extinct. It was the Florida panther, a subspecies, a population that was probably going to go extinct. But it's emblematic also of what happens in domestic animals. Atrial septal defects started showing up. By the time the decision was made to do introgression, almost every single male that was being born only had 1 or 0 testicles. That's not a good sign. It won't last for very long. The incidence of deformed sperm, also not a good sign, was increasing rapidly. Now, because we had also studied other populations, Western and South America, we actually knew that compared with most cats, pumas have really lousy sperm. But the Florida panther is much worse. And then a number of things that aren't necessarily of any evolutionary importance. The incidence of kinked tails and cowlicks and other things you see in lots of populations also became fixed. That's another sign of inbreeding, right? If you see a population of black squirrels, well, they're probably starting to, get a little inbred. So the goals were to reduce the amount and to really, and this is what I want to focus on, reestablish the evolutionary adaptive potential, that key. If you don't have genetic variation, the ability to adapt to new stresses, to new selective pressures, such as a new disease or something, are fewer. And so that's what you really want to try to maintain in populations. Cats were released from Texas, and with lots of work, which makes this study actually very interesting and made it a good model. For instance, at one point, from a domestic cat, a very virulent strain of FELV got into the population, killed very rapidly, almost a high incidence, 60% of the cats for which it came into contact in a very rapid time period. And the vets made the decision around that focus of where the cats were dying. They started catching and inoculating the cats with a domestic cat shot. So the take-home point here is the things we're talking about aren't in a vacuum. It goes both ways. And so it's what I'm really excited about exploring, not just the domestic cat stories, but I think we should all realize that the implications of a lot of what we're going to be discussing go way beyond just the domestic cats, as well as the promise and the potential for the domestic cats can learn a lot from what's going on around it. A couple quick examples even. There's lots to be learned out there, kids. New species of cats are being identified. This was clouded leopard. We identified a new species of clouded leopard just a couple of years ago. New subspecies of tigers being found. Evidence that there's more genetic variation as measured here. There's more, well, this is the number of tigers in captivity versus the number of tigers in the wild and subspecies. Most tigers are now in captivity, and there's more genetic variation of tigers in captivity than in the wild. Understanding genetic variation is crucial to understanding how to conserve this. I've jumped through lots of things. It's impossible for me to acknowledge everybody. These are the folks that are mainly involved in the Florida Panther Project, so I wanted to put that out there. Dr. Steve O'Brien, who I've mentioned several times, and others from the lab over the last few years, involved in almost all of the examples that I've shared with you today. If you'd like to get in touch with me with any question at all, if I can't answer it, I will help put you in touch with someone that might be able to. These are my new email at the Smithsonian. I want to give a special recognition to, if you have not heard of your foundation, the Morris Animal Foundation. In many ways, they are the NSF of animal work. Well worth support, and especially if you're an up-and-coming student or elsewhere and you're interested in any of these things, they're very a good source of a way to get started into the field. So, and they have funded a lot of this kind of work over the years, and I hope they will in the future. And with that, I thank you very much for your attention.